didn't see Sule is here, only Usman. All right, thank you, Dr. Ahmad. So we come again for the number, what number? Number 7, eh?
Number 5.
Number 5. Uh, yeah, I have almost every day Zoom. I just finished one this morning. It's very good. Exchange ideas, exchange knowledge, sharing knowledge on all the issues related. Well, this time we are focused on the biodiversity because Department of Biology supposed to lead, for me, it's supposed to lead in the biodiversity issues. I tried to lobby many universities, many departments to lead, but a lot of restriction with many reasons. Actually, if I can tell you, when we want to work on biodiversity, you only need your brain, your hand, your eyes, your leg, thinking, you can do it. So without money also, you can do it. But unfortunately, I don't know what is the young generation now. They need someone to ask them to do. We need someone to recognize them. They need money, and then they want to do. So we need to change. We need to change. I talked to this idea to many people already, because Malaysia is a very important country, number 12 in the world in biodiversity richness, for example. And then we are very rich in biodiversity, especially now, very hot. We are talking about tiger when we are facing uh, a lot of issues. Number one, ASEAN ministerial conference on tiger. We just had one, and then we are going to celebrate Tiger Year, Chinese New Year, and then we have a lot of issues on flood, deforestation, and so on. There are many other issues related to biodiversity, and this particular thing is tiger. So I think issues of biodiversity is very important. So today we are very fortunate. We have two young scientists who want to talk about. We have two topics here: Medaka and jellyfish, potential tropical marine animals in research. I just got a call uh, from few journalists from the newspaper. They are asking me about the uh, marine ecosystem, right? Marine ecosystem. And then I don't have time to answer because of today, Jumat. I think we can discuss later. I don't know why suddenly they want to discuss on marine. Maybe because of they want to know more, want to learn more uh, of marine habitat, of the threat of the marine life. And if you see in newspaper today, overconsumption and threat to sea life. So this is something that one of the focus on our ocean. We're going to talk about this overconsumption, over harvesting for maybe for the whole year until the World Ocean Day. At least we want to talk about it. So today we are very lucky. So I think we can start now. I give you maybe 20 minutes ish, 15, 20 minutes ish, to present your ideas, your thinking, your activities uh, related to this topic, Medaka and jellyfish. Number one, I would like to invite Dr. Nurul Izza. Um, I don't have to introduce you. Maybe you can introduce yourself along the way, because kita nak santai, just relax, not too serious uh, on our discussion. So we try to bring as many people as possible to discuss later on the issue. So Dr. Nuriza, you can start now.
Assalamualaikum warahmatullahi, and thank you, Prof. Dr. Ahmad Ismail. Okay, right. I shall share my slide. Okay. Right. Okay. So, my talk is about this Javanese Medaka, which scientific name is Oryzis javanicus. Some of us might already know this fish, but I guess a lot of us still do not know about this fish because this is not a very economical important fish. It's just a small fish that abundantly available in our brackish water environment. And this fish is just a small predator that come in lower trophic levels in our ecosystem, but it is a very important species that can tell us about the climate change, and we can also learn evolution from this fish. So brief introduction about this fish. So this fish, we,
Izza,
yeah,
sorry to interrupt. Can you switch on, turn on the camera? We would like to see you.
turn on camera.
Okay.
It's slow lah internet. Okay, right. Right. So yeah. So this fish is a fish that not really gain intention to many people because it is not an economical important fish, but rather it just can be considered as a surplus fish. Okay. Um, However, it's very valuable if we use this fish as a model species to study about the brackish water environment. And this fish is a euryhaline species, so it can be found in fresh water until brackish water, and it is small size. So throughout the world, there are many species of Medaka. One of the famous species of Medaka that has been widely explored and studied is this Oryzis javanicus, or locally known as Javanese Medaka. Okay, whereas the one that I'm talking about today is this species of Oryzis javanicus, which is the Javanese Medaka. And this species, as you can see in this map, it is available in Malaysian water until this Borneo and also this part of Indonesia side. So the distribution of Javanese Medaka is quite wide, okay, throughout this region, where you can see it here in this map number 2. Okay. However, this fish is just recently been introduced as a good model organism to study evolution and also ecological impact in the brackish water environment. And so these are the reason, or these are the factors that make this Javanese Medaka as a good model organism for us to study many biological activities. And if you aware about this other species of fish, which is the zebrafish, that has been used widely to study as a model organism to study a lot of medical related research along with the rodent. So this Javanese Medaka is a species that we can also consider in that aspect. However, this species of Medaka is more suitable to be used to study about the environmental and evolution studies. So this Javanese Medaka is actually more or less a model species that is also useful in the Western and American country where they have this sickleback. So sickleback is also a model species that been widely used to study about the environmental and evolution in Europe and America. So in Malaysia, in Asia, so we can use this Javanese Medaka. So um, a lot of study in Javanese Medaka have been performed to study about the ecotoxicology, some genomic analysis, very preliminary study, evolutionary study, also in preliminary with comparison to other Medaka species, and also its adaptive physiology. So all of study in this Javanese Medaka are still in infancy because it just been recently introduced as a species of interest in many biological study. So these are some of the fascinating Medaka, especially Japanese Medaka that already being manipulated through genetic study to make they become more fascinating for further study and also as pet fish. For Japanese Medaka, we still haven't managed to do this thing yet. Okay, this is all for Japanese Medaka. So basically, what we had did in Malaysia in terms of Javanese Medaka, with the preliminary study, we already tried to determine the sex of this fish using the molecular approach. The simplest way and easiest way is that we can identify this fish using the phenotypic or the morphological features of the fish, where there is differentiation between the male and female. And one of the most distinct features is that female have this branch and a fin, while male doesn't have any branch and a fin. However, for some people that don't really familiarize working with Medaka, to differentiate this male and female fish of Medaka is quite tough. Okay, so therefore we want to go further to help those people that do not really familiarize with this Javanese Medaka, so that they can use this fish as border species, because we know that whatever activities, whatever effect or factors, there will be different between male and female. So therefore we start to study this molecular identification of the sex of the fish using molecular method. From literature, we know that male of Javanese Medaka carrying ZW chromosome and it's reside in chromosome number 16, whereas for female, they carry the ZZ chromosome formation in chromosome number 16. And in our study, we had managed to amplify using a primer that possibly as a primer of this DMRY gene that determine the female and male of Javanese Medaka. From our study, we found that the primer that we use that relate to this DMRY gene could differentiate the male from female, whereby the male have two bands producing under the electrophoresis, whereas female only have one band. So with this, we can help other people that do not really familiarize with Medaka to look for its male and female using this molecular technique. And then we also have this genetic diversity study that we performed quite recently with selective population, but this is going to be expanded further in the future. So recently we only selected these three different population of Medaka. One is at Kuala Triang, which is at Langkawi Island, and then one is at Kuala Perlis, and one is at Bagan Lalang. So the three different places here were selected randomly based on different features of the places. For Bagan Lalang, so it is a place with high aquaculture activity. For Kuala Triang, so this is an island population of Langkawi that's considered as a place of tourism. And then for Kuala Perlis, so this is where we collect the fish near the residential areas with paddy plantation. And so from our study, when we screen two SSR primers, and we found that the allele frequency pattern diverse in Kuala Triang population compared to Kuala Perlis and Baganalang. And the heterozygosity value also support the finding of our allele frequency study, whereby it said Kuala Triang have high heterozygosity value. And when we look at the inbreeding levels from the two SSR markers that we use, unfortunately, all of the three population that we studied in this case, they were highly inbred. So for the high level of frequency of allele that we found in Kuala Triang with high level of observed heterozygosity, so this telling us very important things that when there is high heterozygosity, so there is high genetic diversity. And when there's high genetic diversity, so there will be high potential for the population to get evolved and adapt to the local environment change. Whereas when we look at this decrease in the heterozygosity and also in less variable allele frequency in Baganalang and Perlis, it tells us about this probably present of this Solahudin effect, that telling us that when this Medaka fish had been isolated according to different population, they might have this hindrance to travel from one place to another due to the geographical barrier. And when this happens, the gene flow will be stopped to move from one population to another, and it will introduce the genetic drift in the subpopulation. From our further analysis to see about the population relationship or the population similarity, we found that Kuala Perlis and Kuala Triang population are closely related compared to the Bagan Lalang populations. And so this is in correlation and agreeable with the geographical location of the fish where we collected. Okay, where here, if you could see in this map, it tells us that Kuala Triang is close to Kuala Perlis, whereas Baganalang is distant away. And when we look at one of the locus, we found that there is a probability of mutation that could happen in the Kuala Triang fish, whereby it's introduced the possibility of increasing the allele frequency and also genetic diversity in the population. If you could look back at here, the Kuala Triang has this high variation in its genetic allele frequency and also in its observed heterozygosity value. Okay, yeah. So then that's when we go for that population genetics study to understand about the evolution of how the Medaka spread and what will be the factor that might contribute to the spreading of the species throughout Malaysia Peninsula. After that, we also want to go further to focus on one of the physiological aspects that important in this Medaka, which as a urihaline species, so it has to have this ability to osmoregulate. Okay, and there's quite abundant of genes that have been studied relate to this osmoregulation. And in this study, we select one of the genes, and we use this to look at two different populations. One is at Kuala Sepetang and another one is at Sungai Pulai. The one at Kuala Sepetang is a conserved area, which is at the Matang mangrove, whereas the Sungai Pulai is the one that we collect from the river, nearby the river that's surrounded by industrial activity. So from our study, we found that the gene that we focus on, which is the AMD gene, showing there's some variation that present in the Kuala Sepetang population, but in Sungai Pulai there is no variation at all. And therefore, we select some of the individuals in the Kuala Sebatang and Sungai Pulai to go further to look at what would be the possibility that telling us about this diversity that we observe in the Kuala Sebatang and no diversity that we observe in the Sungai Pulai. And when we do further analysis, we found that the probability of the diversity that we found in the Kuala Sebatang may be due to there's differences in the nucleotide sequences, whereas for the Sungai Pulai, the sequences is not that differentiate from each other individuals that we studied. So from our study, when we look at this, we conclude that these AMD2 genes that is involved in the gill that already know in the gill of oryzis platypus to play a role of sodium excretion into hypertonic environment and inhibit the kidney of oryzis platypus is present in both population of Oryzis javanicus in Sungai Pulai and Kuala Sebatang. However, there's variation that we could found in the Kuala Sebatang population. And when we found the diversity of variation in these AMD genes in the Kuala Sebatang, so this suggests that the population in Kuala Sebatang is more plastic than population in Sungai Pulai, which is telling us that the population in Kuala Sebatang have high potential to evolve and ability to adapt to environmental change compared to the one that we found in Sungai Pulai. Furthermore, this is for a long-term study when we want to target this Javanese Medaka as a good model organism to study ecology and evolution. So we want to go further in the future in detail to look at the QTL and also the genome-wide association studies. From our collaborator, we had luckily, because our collaborator from Japan, they had managed to sequence the whole genome of the female Javanese Medaka fish that comprise of 24 chromosomes. And from the data that our Japanese collaborator had, we had managed to design SSR markers, and these markers will be in the future used for the further QTL and genome-wide association study. These are just some example of the 2 chromosomes out of the 24 chromosomes that had been fully sequenced in the female fish of Javanese Medaka. For chromosome number 1, the total size of the chromosome is 37 million base pairs. For the chromosome 16, it is 35 million base pairs. So why here I'm showing the chromosome 16 straight away after chromosome 1, it is because I just want to show you that this is the chromosome where there's a sex gene reside in Medaka, and this is the chromosome where there is non-sex gene or autosomal gene. So from this chromosome 1, we found mono, di, tetra, pentahexa, and complex SSR markers, SSR repeat motif, and then from there we design several markers. And here I just want to go in detail in the di nucleotide, where here in this chromosome number 1, majority of the dinucleotide we can found as AT composition, whereas in chromosome 16 it is found as TG. And I highlighted here this CG because CG is also known as the CBG island, which is the position of the genome that hardly methylated. And usually in this CBG island, there is methylating activity of cytosine with sometimes it can cause a gene expression change. And this CPG island is also a target region in many epigenetic study. A lot of study had been performed in other species in the CPG island, where we want to understand about cancer, where we want to look at the reduction and oxidation, and also at the transposable element. And here, usually at the CPG island, when there's mutation, sometimes if they are upregulated, they can influence the expression of the genes. So in the chromosome number 1, we found that there's only one SSR marker, so there's one potential diversity in this CPG island in the chromosome number 1, whereas in the chromosome number 16, there's 3 potential sites, whereas there's diversity in the CPG island that can be potentially used to study further about this other epigenetic event that probably could occur in this sex chromosome. Whereas the GC, so GC is also in the CPG island, but GC is usually being found in the down-regulated part of the gene, whereas for the CG is the one that we usually found in the upregulation part of the gene expression. So this the in progress that we currently still undergoing study, where we still want to expand further to include more population of Javanese Medaka in Malaysia, so that we can have more information about how the distribution, the evolution, and also the ecological impacts that might tell us about the behavior of this fish throughout the Malaysia. And also we want to explore further about the osmoregulation genes also in Medaka using different population. And we also have some lab-based study that we currently use to prove evolution related to ecology and anthropogenic factors. So with that, I would like to say thank you to a lot of students and also to a lot of lecturers that introduced me to this fish, and also with our Japanese collaborator, especially from Dr. Ahmad Ismail, who first introduced me about this fish. With that, I end my presentation. Thank you.
Okay, thank you, Dr. Nurul. Very good. 20 over years ago, when I talked about Medaka in Malaysia, they said no commercial value. But when we talk about the testing organism, there's a lot of things that we need to do, because the demand will be high one day after public are very concerned about this, about the safety of their products, for example. All right, don't have to discuss more on that. We can discuss it later if we want to know more. Thank you, Dr. Nuriza. And next, Dr. Amal, we call upon this Dr. Syazwan, Muhammad Syazwan, because another one here, Siti Syazwani, saya tak call Siti Syazwani. So thank you, Dr. Amal. This time you bring 2 young scientists to talk about the future of marine or coastal life. That
I have
we will
question.
promote more later. Ya, apa questionnya? Tunggu kemudian, nak tanya sekarang ke?
Okay, dari hujung.
Right. Dr. Wan Muhammad Syazwan, graduated from the Malaya, and then I give you 20 minutes to present your, to share your information. Dr. Muhammad Syazwan.
Okay, Assalamualaikum. Thank you, Prof. Mike. Let me share my screen. Okay, everyone can see the screen.
Yes.
Okay, go
Alright.
ahead.
Yes, doctor.
All right, thank you. So, Assalamualaikum and a very good afternoon to everyone. Firstly, thank you, Prof. Mike, Dr. Amal, and everyone involved in organizing this webinar for inviting me. And welcome to everyone. Thank you for your time. I also can see our KJ here. Dr. Syazwan is also here. Right. So, without further delay, let me start my presentation. So, basically, the topic of today sharing session, basically this is only a sharing session lah. It's a casual talk, kita sembang-sembang saja, like Dr. Prof. Ahmad said. So basically, my topic is on the jellyfish science in Malaysia in terms of the challenges, the prospects, and as well as the new discoveries. So basically, this is actually a part of my PhD study. So, right. So, firstly, what is jellyfish? Do everyone in this room knows about jellyfish? I'm pretty sure that every one of you have, at some point of your life, have encountered jellyfish, or at least heard about jellyfish. So basically, jellyfish is an organism that we would rather consider as ancient, because they have lived in our ocean since 5 million years ago, right? Even before the dinosaur time, right? So, a quick introduction for jellyfish that is relevant for today. First, that you need to know that they are very watery, they are gelatinous, right? Their body composition consists of 95% of water. And another thing that is important and always cause misunderstandings for the public is that jellyfish is not a fish, but jellyfish is actually a type of plankton, okay? If you see the SpongeBob SquarePants, right, and you can see the villain in the story, it is actually a real plankton. And another thing that you need to know about jellyfish is that some of jellyfish, right, some species of jellyfish, they are being harvested, okay? They are being consumed by human all around the world, not only in Malaysia, all around the world. And Malaysia is actually one of the exporter, the major exporter of jellyfish food products in the world, right? And the next thing is, I think everyone knows that jellyfish has stings. When we say about jellyfish, everyone always say that it has painful stings, sakit, and then there are many cases of deadly cases of jellyfish stings. But not every species actually are dangerous. Not every species can cause death, right? And lastly, you need to know that a mature jellyfish, adult jellyfish, it can lay eggs about 45,000 per day. If you compare with fish, right? Fish only can lay, if I'm not mistaken, about 100 to 1,000 eggs per day only, right? So, the term that I want to introduce, for those who still doesn't know, is today is the jellyfish blooms. So what is jellyfish blooms? Okay, so basically jellyfish blooms is a natural phenomenon where there is a sudden increase of population, or the numbers or the density at certain times and places, meaning that there are certain sudden increase suddenly. So we also call the bloom sometimes as a swarms, sometimes also aggregations, and the numbers of the jellyfish in a place sometimes can go to millions, as you can see in the picture and also in the video. All right. And however, the thing about the jellyfish blooms is it's not a weird phenomenon. Actually, it is a rather natural phenomenon. Right? It happened since million years ago, but interestingly, since 30 years ago, the annual events of jellyfish blooms has significantly increased. And the increase of jellyfish blooms is not only happening in one place, but it occurs in marine ecosystems worldwide, serentak. So it's very interesting. As you can see in the slide, we have cases in Japan, in Australia, in USA, India, Philippines, and we also have jellyfish blooms in Malaysia. As you can see in some states, in Pahang, in Penang, in Labuan, in Kelantan, Kedah, Langkawi, and Terengganu. Okay, and the reports of jellyfish blooms in Malaysia is not. I would say that it attract a media attraction recently, because I can say that there are many media start to report the cases of jellyfish blooms in Malaysia, and also in the social network, right? From my study and my literature review during the PhD study, we have compiled about at least 45 cases of jellyfish blooms from 1960 to 2010. Okay, So the jellyfish blooms actually is not new in Malaysia. It happens and has been reported since the 1960s. So this issue of jellyfish blooms, it has triggered some questions, right? Why? Why is the bloom happening? Okay. But more importantly, what are the diversity and distribution of jellyfish in Malaysia? Okay. So the blooms are related to their feeding habit and reproductive pattern. Are these two ecological components, ecological aspects, the feeding as well as the reproduction, are actually causing the blooms? And more importantly, what are the impacts of jellyfish blooms to the ecosystem, to the human, and also to the animal in the coastal waters? So, however, unfortunately, the baseline diversity and ecological data of jellyfish is seriously lacking. Because if you can see, if you take a look back at the previous jellyfish studies in Malaysia, you can see that basically the most records or reports of jellyfish is actually from the non-taxonomical studies. And these studies actually study on the envenomation, on the toxicology, on the fisheries, right? None of the studies are actually studying the morphology, the taxonomy, and also the molecular of the jellyfish. And also many of the reports are actually comes from the non-academic sources. For example, the media, the field guide, the encyclopedia. And because of that, many identifications in these resources are very doubtful. Some of them, actually majority of them has been misidentified. Not only that, they were poorly described. And this is very unfortunate for our science, because there is no, for most of the species in Malaysia, there are no voucher specimens, right? So in terms of the previous study on the ecological aspects of jellyfish, there are very few population and abundance studies. Some of the studies have been done in Penang, for example, in Perak and even in Selangor. However, these studies only are done at a very short-term period, okay, normally less than 6 months, and they did not try to relate the reports of the jellyfish, the occurrence of the jellyfish in the area with the ecological aspects in the area. And none of the coastal food web studies in Malaysia pertains to jellyfish, meaning that when scientists try to construct a food web study, construct a food web in any coastal water, for example, or in the estuary, they did not incorporate jellyfish as one as the consumer in the food web. So this will cause the misinterpretation of the trophic interactions. So picking up from that, so during my PhD study, we have conducted a scientific project entitled Diversity and Ecology of Jellyfish in Malaysian Waters. So basically, the members of this project is from UPM, right? Because I am now from UPM and also from University of Malaya. And this collaboration is ongoing. So the objective of the study is, we have two objectives basically. First is we want to update the knowledge of jellyfish diversity in Malaysian waters. How we do that? We try to do the field sampling throughout the coastal waters in Peninsular Malaysia. As you can see, this is the numbers of this is the location of the sampling sites across the Peninsular Malaysia. And secondly, we pick a site, particularly in Peninsular Malaysia, which is the Klang Strait, to study the environmental, the biological, as well as the ecological role of jellyfish in the study area. So you may ask, why we choose Klang Strait? Because actually Klang Strait is the important ground for the commercial fisheries and also for the jellyfish fisheries. Selangor is actually one of the major producer of jellyfish fisheries in Malaysia. And secondly, because of the Klang Strait has the most extensive mud flats, dataran lumpur, and also the mangrove, hutan bakau in Peninsular Malaysia. And third one is because the area experienced the heavy human anthropogenic impacts in terms of the coastal pollution, in terms of the power plant operation. We have a power plant at the Klang Strait. And also because of the shipping ports, we also have Pelabuhan Kelang at the Klang Strait, right? So it is hypothesized that because of this environment right? Because of there is the interaction between the environment, the biotic factors, the anthropogenic factors, the human-related pollution, the eutrophication, and so on. Actually, all these conditions promotes the jellyfish blooms. So that's why we try to tackle the environmental aspects, the biological aspects of jellyfish at the Klang Strait. So this is just want to show our field survey. How did we collect the jellyfish across Peninsular Malaysia? We try to collect them using different methods. For example, we did the beach survey. We also collect the jellyfish using the scoop net, and we also collect the jellyfish using the fish net. The thing about jellyfish are some of the challenges in collecting and also in handling the samples from the water is because jellyfish, it is not easy to target, meaning that they are highly seasonal. They are very difficult to find in water. So if you compare between these three methods, right, the most successful method is actually by using the fisherman net, okay? Because the fisherman net, for example, in Klang Strait, they set their net across the tidal current. So the jellyfish just being trapped in the net when they are being drifted with the tidal current. So that is the most successful method. Okay, this is the actual picture of me going to the field. This is the boat that we have been used. You can see that the jellyfish, when they are blooms, there are too many of them until actually the fisheries resources, the other fish resources that should be collected by the fishermen is gone. As you can see in this trap, there is a hole in the boat where they collect all the fishes, the shrimps from the nest. They will collect it in the hole. But you can see during the blooms, right, there is no longer fish and shrimps. It is being preoccupied by jellyfish. So this is very, I can say interesting, but actually it is actually impacting the fisheries in the Klang Strait itself. I will talk about it later. Okay, so this is basically the laboratory procedures that we have done during our study. So this is the standard one, like we always do for the fish sampling. First, we collect the animal, we collect the jellyfish, and then we measure them, we weight them, and then we try to identify them, and then we take pictures, right? And then I want to highlight that during the sample processing, the two things that are very important is we are trying to assess the feeding ecology by looking at the stomach content analysis, as well as the reproductive biology by looking at the gonide. Okay, if you try to compare jellyfish with other fish, for example, let me pick fish because Dr. Izza has mentioned about the sexual characteristics of fish, where you can identify the male and female by looking at the fin. However, there is no such secondary sexual characteristic for jellyfish. It is very, I don't think people can identify whether a jellyfish is a male or female by just looking by naked eye. So what we need to do, we need to dissect the jellyfish, we need to take out the gonad and put it under the microscope to see the to define the sex and also to define the maturity status, right? And lastly, we preserve the sample. Okay, so moving on to the next part, where I will show you some highlights of our findings and some new discoveries. And basically, this can be categorized into 4 parts, which is in terms of the diversity, the distribution, and secondly, in terms of the community structure. Next is the reproductive pattern, and lastly, in terms of the feeding ecology. So, in terms of diversity of jellyfish in Malaysia, from our present field surveys, we have, let me minum air jap, mulut kering. All right, for the diversity of jellyfish in Malaysia, so from our field survey, we have collected 10 species, which is, okay, this is the picture of the 10 species, 9 of them is shown here. And then 2 species are the first record in Malaysia, right? It has never been reported before. And then from our Okay, I forgot to mention that the way that we try to update the diversity of jellyfish, there are two ways. First, we did the field sampling. Secondly, we try to look at the past records through the examination of the water specimens in the museum, as well as we were looking at the literature, try to find and cross-check and examine, verify whether the species are valid or not. And from that, when we combine the field surveys as well as the past records, we conclude that there are a minimum of 20 identified species in Malaysia. This is only for the class Kyphozoa of jellyfish. I'm not talking about the other class for jellyfish. This is our class of jellyfish that were selected in the study because of their abundance, right? And there are other 9 unidentified species. We only managed to identify until to the genus level. And there is 5 species from the records. They are still unverified until today whether they are actually in our water or not, right? And all the 20 identified verified species, this is actually comprised of about 9% of the total global species richness of Scyphozoan jellyfish in the world, all right? So throughout our examination, throughout our updating of the species checklist, we have made some suggestion in terms of we want to standardize for future researcher about the identification of some species. Okay, this is the species, just to show you that this is the species that have been mistakenly identified, and this is the updated or corrected species name. Okay, and interestingly, half of these species is actually the species that has commercial importance. Okay, so it is very unfortunate lah, because even in the official fisheries statistics of Malaysia from Department of Fisheries, they did not manage to identify the jellyfish. So it is a very it's not good for the especially for the fisheries in terms of identification the species, which species, and also the ecological of the species, right? And, okay, I try untuk lajukan Rahmat. So in terms of distribution, I just want to highlight that in the West Peninsular Malaysia, we have the West and also the South. We have the most species of jellyfish compared to the other parts of Malaysia. And in the West and South Peninsular Malaysia, we found that actually the jellyfish are very abundant in the mangrove and mudflat habitat compared to the other type of coastal habitat. Why? Because this type of habitat, the mangrove and the mudflat, they serve as a nursery, as a feeding, and also for the maturation grounds of jellyfish, right? So in the cluster rate, what I can summarize from these 2 graphs is that jellyfish in the cluster rate in the mangrove and mudflat, they are highly seasonal. Okay, they in terms of their seasonal distribution, right? They are more abundant during the wet period, specifically during the northeast monsoon compared to the dry period during the southwest monsoon. So they prefer the northeast monsoon because we found that the jellyfish their abundance is correlated with the decreased temperature, the decreased salinity, decreased DO, as well as the decrease of pH level. And we assume that the increase of jellyfish during the northeast monsoon, during the monsoon timur laut too, it is because of the increase of food abundance during that time. And we also try to assess or tackle the reproductive pattern, and we found that the jellyfish actually, they spawn, they spawn throughout the year. They have continuous spawning pattern, right? And this is specifically for the tropical one, and it is very different compared to the temperate, the species from the temperate water, for example, in the Europe, right? In Europe, they have their seasonal pattern. The reproductive pattern is actually depending on the season. For example, they spawn during the summer, and then they keep dormant until the next summer. And in terms of the growth analysis, we found that in terms of the growth, right, their growth and total mortality is about the same with other regions, with other populations. However, in terms of the lifespan, the jellyfish in Malaysia actually has a longer lifespan compared to the other places, right? So in terms of their feedings, we found that jellyfish in Malaysia actually are they feed mainly on the zooplankton, especially the copepods. And we found that within the cluster itself, we managed to cluster the species into 3 different trophic guard, meaning that there are 3 different feeding habits of jellyfish within that area only, within one population of jellyfish. So there are 3 feeding habits that we managed to identify, right? So during our study, we try to incorporate jellyfish into the food web. Like I mentioned before, in Malaysia, the food web studies, they have never been include jellyfish. So now we try to put jellyfish into the food web, and this is what we found from the stomach content analysis, as well as from the stable isotope analysis. We found that jellyfish actually positioned at the 3rd trophic level in the food web, and they consume. Okay, so the thing about the Klang Strait and also other coastal waters in Malaysia, mainly the primary sources are the mangrove, the phytoplankton, as well as the benthic sources. We call it as a microphytobenthos. And interestingly, we found that jellyfish actually get their trophic energy from the phytoplankton as well as the microphytobenthos in the benthic area. But there is very little sources of mangrove. Very interesting, because actually the Klang Strait has is fringed or bordered by a very long mangrove habitat along the coast. So it is very interesting to know that. And so what can we say about this diagram? So actually, jellyfish, they consume these 2 sources, the phytoplankton as well as the microphytobenthos, indirectly. Okay, they did not consume it directly, but indirectly. How? Through their consumption of the copepod, right? So basically, the phytoplankton and the microphytobenthos is being consumed by the copepod, and then the copepod is being consumed by the jellyfish, right? So in terms of the food web, we only have managed to resolve this question. But if you take a look at the right side of the diagram in terms of their relationship, in terms of the energy, the trophic level, the trophic interaction with the higher consumer. For example, the fish, the seabirds, or other animals, and also for the human, we haven't managed to study that, right? And we leave it for the future studies, right? So moving on to the prospects and future studies, this is going to be a very quick one. But I think this is very related to ones that are raised by Prof. Mike, because until today, despite of the baseline data that we have provided throughout the study, we still cannot understand why the jellyfish is blooms, okay? Because in other parts of the world, scientists have suggested some hypotheses why the jellyfish are blooms. First, because of the coastal pollution. Next, because of the overfishing. Okay, next, because of the climate change. This is a very interesting topic. And lastly, because of the increase of human-made infrastructure. Because jellyfish will lay their eggs, and the eggs will attach the substrate on the human-made infrastructure. So because of that, the jellyfish is increased because they are now mainly human-made infrastructure at the coastal area. But to date, we still can't resolve this question. And the next thing that I think that very important to be explored in the future studies is in terms of assessing as well as mitigating the negative impacts of jellyfish blooms. Okay, let me show you what are the impacts of jellyfish blooms. Firstly, they cause sting threats to human. Why? Because they have stings. This is the picture of the stings. These are real cases in Malaysia, in Kontra Kinabalu. This is also in Sabah. This is in Sabah. Mostly are from Sabah, from the media. But actually, we also have many cases, unreported cases from my interviews, especially at the fisherman village. Biasanya, normally the fishermen, they always get stung by jellyfish, but they didn't report it. They didn't report it, so media didn't report it, didn't record it. So there is no record of the stings, but actually the case is higher. Okay. So the next thing about the impact, the negative impacts of jellyfish blooms is it will affect the tourism. When they are blooms and because of the stings, people are afraid to go into the water, and it will impact the tourism. Okay, the third one is very important in terms of the ecosystem and also the socio-economic. Because of the blooms, it has become a threat to fishing sector. This is the real picture in Japan. It also happens in Malaysia, as I showed in the boat that I have used to collect the jellyfish. You can see that because of the during the blooms, fishermen cannot collect their normal fishery sources, and in some cases the boat has been capsized, meaning that the boat tu dah terbalik karam because the jellyfish is too much, sangat berat, and the boat cannot tampung the biomass, the weight of the jellyfish. And because, the fourth negative impact is very related to the last part of my results, where I showed that jellyfish is actually important zooplankton predator. So they feed on zooplankton. Okay, the thing about zooplankton consumer in the food web, especially in the Klang Strait and also in other regions in Malaysia, in the Klang Strait, about 50% of the fish actually also consume zooplankton. So if the jellyfish is blooming in the Klang Strait, so you can imagine that the zooplankton sources in that area will be gone, will be reduced. So what happened to the other commercial fishery stocks? So it will cause the reduction of the commercial fishery stocks because of the competition with the jellyfish for the zooplankton. Okay, the last thing, the impacts in the Klang Strait, for example, is the blockage at the coastal power plants. Okay, you can see that there is a power plant right there. We have a Kapal power station, and normally because of the blooms, the jellyfish will get ingress into the screen of the power plants, and this is the picture. You can see the tentacles is hanging down, and in some cases, for example, in Perak, if I'm not mistaken, I don't remember the name of the station. There has been what we call it, the power disruption of the power station. And what happened? The nearby town of the power station, all of the houses, the shops, they are out of electrics because of this issue, and it has been reported it has caused a loss about millions because of that, millions of ringgit because of that, right? So the next thing about the jellyfish blooms is it not only causes the negative impacts. Okay, you need to remember, you need to know that the jellyfish blooms also brings economic benefits to fishery. In Malaysia, we commercialize jellyfish. But interestingly, although all around the world, people are trying to collect jellyfish, the fisheries of jellyfish is increasing. But in Malaysia, I don't know why, from the records of fisheries statistics, I can see the decline of fisheries jellyfish catches throughout the year. And for some jellyfish ground, for example, in Sabah, there is no longer jellyfishing in Sabah. Dah tak ada dah jellyfishing in Sabah now. But 10 years ago, yes, there are. So that is the example of the situation that we need to tackle. We need to understand more. Why is it happening? And because people are harvesting jellyfish for two reasons, mainly. First, because of the food source to human, and secondly, because of the raw material. Okay, for the food source, this is very straightforward. You can see that jellyfish are being used as seafood products, and also for the sushi. If you go to the sushi places, you can see that jellyfish is one of the menu. And lastly, to the raw material for the medicinal products, for the cosmetics, for the fertilizers. The thing about this is there is very minimum or lacking study that really investigate the level or try to assess whether the food that they are consumed, the jellyfish food too, is actually safe or not. There is no risk assessment that has been so far, right? So I am going towards the end, right? So just want to highlight, this is the collaborative research that has been done in Malaysia. Okay, for the last 2 years, we have the IOC Westpac from the UNESCO. And this is actually a collaborative research from different countries, from Malaysia, from Bangladesh, from Thailand, Indonesia, Philippines, Vietnam, and China. And in the local scale, we have UPM, we have UM, we also have USM, UKM, and UMT, as well as the Department of Fisheries. So with that, I end my presentation, and I open to the questions to the floor. So that's it.
Okay,
Thank
terima
you.
kasih Dr. Syazwan, very good. This is what we discussed long time ago. Never end because not many people, number one. Number two, monopolized by certain people. So it's not that many are willing to open and share with others. You put the JSPS there until now they continue. But I hope and JSPS have a good leadership now to bring more people in. And you are very lucky working with the University Malaya because in Malaya actually they are the one who lead in the JSPS program. They should, I don't know now, but before the chair is at their place. Uh, Anyway, don't care about anything sponsorship or whatever agency. The most important is what you are doing and try to answer the problem or issues of jellyfish in the coastal environment. There are many aspects that we need to look at. Uh, right, never ending, never ending. This is, but we like to change topic. We like to follow the grant. That's why our development and research is very slow. Slowed by, I always say by the leaders and the panels. So sometimes they look what they like. They don't see the overall picture of biodiversity in Malaysia. All right, Dr. Amal. Time dah habis tapi kita bagilah peluang Dr. Amal nak tanya apa. Lupa
Assalamualaikum.
tidur dah. Okey, bangun lagi.
Dah nak hujan dekat UK dah.
Dekat mana ni ada bunga-bunga ni?
Taman
Oh.
Ahmad.
Alright, okay.
This question actually for maybe for Dr. Izza or Prof. Ahmad. We can see here that a lot of study now they start using Medaka. Okay, we should promote lah Medaka because that's from our country, native to our country. But at the same time also, we can see a lot of people still prefer to use zebrafish, which is a competitor for the Medaka in research. Several problems why people still using zebrafish because zebrafish can produce a lot of eggs. Okay, compared to Medaka. One more thing because they're easy for them. Just buy it from the supplier. How much you want? Just pay money. Not like for Medaka, you need to go to the breakage area, collect one, bring to the lab, condition, make sure it's free from disease, from parasite and everything. That's actually problems, constraint. That's why scientists not really prefer to use Medaka in this country as compared to the zebrafish. Any comments on that? How to promote that, Promat and Dr. Izza?
Dr. Izza, komen dulu. Mana dia?
For me, if we want to go further, we might as well as try to establish a lab population of our Medaka as well as how they did for this zebrafish, where they have this lab population. This genus of fish for Japanese Medaka or Rhizius latipes, they already has this lab who established the lab population of Rhizius latipes, and they being supplying the eggs and the culture of the Javanese Medaka to other countries. So we can do that possibly for our Javanese Medaka so that we can also in line with the zebrafish that people are most fond of in Malaysia currently.
But thank you. That's the good idea lah. Maybe that's gonna be the opportunity for us. Okay, establish the Medaka and then can promote that lah later on. Right, thank you for the comment, Dr. Izza.
Okay, so the most important is attitude. Sometimes we are thinking of experiment, not research, right? So this is the thing that it's about time. All the young generation, I create a gap so that no interference from those seniors like what happening to me when, as I mentioned earlier, when I proposed 20 over years ago on the Medaka, they turned down because they said there's no commercial value. no interest for the country. So what happened now? Say for example, you want to talk about food, talk about cosmetic, they need to test their toxicity and so on, and they need a very good or indicator. That's why Dr. Izza is doing on genetic. Even in Japan, when they are doing on Medaka or Taiwan, Hong Kong, they don't know the background of the fish. So when we comment about the background of the fish, and they start coming to Malaysia looking for fish. So this is something that they cannot do it because when they keep the fish in their country, they need to study the genetic. So finally they come back to Malaysia to collect the fish. So the potential of collaborating or using our laboratory is very important. That's why there is one Singapore company using zebra fish finally changed to Medaka. So and we are still bringing back all the tools and experiment that we learned from overseas to our country, and actually it's good for them, not selling their equipment but selling their animals too. This is something that we need to look after. Another thing was the transgenic fish. I don't really keen to bring it to Malaysia because our Malaysian people are not disciplined enough. The Tamal have enough story on invasive species. We don't have disciplined people in Malaysia. So even though some people try to bring in, but I suggest not. And the quarantine department in Malaysia have to monitor this. Okay, I open to one more question because we are late 15 minutes. Anything else? Dr. Wan Syazwan, you have to develop more people to monitor quickly on jellyfish.
We need more people, Prof.
Not just in the, say for example, in Langkawi, it's a very strategic place to monitor, and then the life cycle, the nutrient, and many other things that people cannot answer. And when we have the case, the journalists will look at the same people, and the same people will give the same answer, and every year, you know. So when I give a different answer, they will ask me again, is it right? Because someone said this. So based on research, and then based on new funding, we need to upgrade the knowledge to the public. All right. So if no answer, I would like to thank all of you. We do it every month. Thank you, Dr. Amal. Don't stop. We keep promoting young people, not only in Jabatan Biology UPM, but we try to search from other universities also. I just got the message from UTHM. They miss again this this time. I said, well, it's okay. Next time you become the speaker, maybe you don't miss the Tama. We look for the better topic. One more response. All right. So thank you very much. I'll see you again next week. Hopefully our discussion today will continue and continue. So finally we'll bring more people looking at our biodiversity. And before I forgot, the Tamal didn't remind this. I just sent to many those who work on biodiversity. I'm collecting articles now on the assessment of biodiversity. It's too busy to write to everybody. Tama, you need to help me. We need at least, I don't know how many